Unit 5 focuses on heredity and the biological concepts and processes involved in ensuring the continuity of life. What is continuity of life? Well, you have to look at reproduction. You have to look at survival and reproduction and what that's really going to mean. Now, asexual reproduction is obviously not using gametes, or at least the union of gametes, and therefore only one parent produces offspring that is genetically identical to the parent. This variability or this lack of variability can be very useful when you're looking at a non-changing environment, but is fairly limited in a changing one. This could be something like the budding of a hydra, the vegetative propagation of a redwood, or parthenogenesis, which is when you have females that are capable of reproducing without sperm. It could be the whiptail lizards or the queen bee producing the drones. Either way, asexual reproduction does not create genetic variability, but it does create organisms and can be very good for quick reproduction in a non-changing environment. Bacteria also asexually reproduce through the process of binary fission. You're still going to have chromosome replication, but that circular chromosome is going to be split into two, and then you're going to have something that looks a lot like cytokinesis dividing the cells. So this binary fission process is probably the oldest form of reproduction that we know of, and is clearly the precursor of meiosis. Well, if bacteria reproduce through binary fission, how do they evolve as fast as they do? Just because the bacteria reproduces through binary fission does not mean that they don't have sources of recombination and variation. When you look at the actual chromosome of the bacteria, it's this long loop folded structure that you see in the diagram. But you also have these small loops of chromosome or plasmids. So all of that's going to work together to enable change. Transformation is when bacteria uptake genetic information from their environment, be it something that they can incorporate into their chromosome or plasmids that they can take in. Transduction is going to be when bacteria are passing information between each other with the process of a viral intermediary. So a virus infects one bacteria, picks up genetic information, and then when it infects the next bacteria, part of the other bacteria's genome becomes part of the new bacteria. Or you could have conjugation, where you use the F plasmids, the fertility plasmids, to enable the creation of a sex pili and exchange genetic information directly. So just because something reproduces asexually doesn't mean that it can't create variation, and bacteria are very good at that. When you move on outside of bacteria, you move on to something like an animal cell, you're going to see differences in the life cycles from organism type to organism type within eukarya. Human life cycle, we should have sort of at least a passing familiarity with. You have the egg and the sperm that are produced through meiosis. Fertilization brings them together. Those haploid sperm and eggs become diploid zygotes that then goes through mitosis and development to grow into the adult form. So humans, we see our chromosome map here. We can see within this picture that we have an X and a Y chromosome. So we're looking at a male in this karyotype. But humans are just an example of a life cycle. They're far from the only one. Sex determination is a key part of this because the XY chromosome that we're used to thinking of as a male in humans is far from the only way that sex can be determined. For example, you can have an XX male and an XY female. It all depends upon the presence of a particular gene, the SRY, the sex determining region of the Y. Even if you just look at the chromosomes, we'll find that in different types of organisms, it's not the same. So in chickens, it's the female that is hemizygous for one. So you have the ZW and the ZZ. In bees, you have the females that are XX, the queens, and then you have males that can be XY or X. So we can see the different sort of sex determination there. And in grasshoppers and locusts, the XX female and the X male, there is no Y chromosome. There is no sort of relationship there. So sex determination is different from organism to organism. Mitosis was a non-reductive process, so the cell never altered itself from a diploid relationship. But in meiosis is a reductive process. The goal is to produce haploid cells with unduplicated chromosomes. So you have interphase, just like normal. Those sister chromatids are going to form. You're going to have the double-stranded chromosomes. Those homologous chromosomes will separate in meiosis 1, and then you will have the sister chromatids separating in meiosis 2. So the process is very different in meiosis, but the key is that it is a reductive one to produce haploid gametes. In meiosis 1, the homologous chromosome pairs separate. So we find that the maternal and paternal copy of each chromosome come together in prophase 1 as a tetrad, as a set of 4. They overlap at the chiasmata, but everything else we see is pretty much the same. The spindle fibers form, the nuclear membrane breaks down. But in mitosis, all of the chromosomes separated independently, and that is not true in meiosis. So the chromosomes are going to align in the middle at metaphase, the sister chromatids are going to remain attached, and the homologous chromosome tetrad is going to split in anaphase 1, and then telophase 1 will reverse the prophase process, and then we'll go into cytokinesis. Technically, the one in the middle here is called interkinesis because it is an intermediate form, but you get the idea. Meiosis 2, the sister chromatids separate. So this is just like mitosis. You're going to go from sister chromatids to single chromatids. Prophase, metaphase, anaphase, and telophase of mitosis, they look identical. The only difference here is that A, it's happening in haploid cells because there's only half the chromosomes, and it is occurring in 2 cells at once. So you had a tetrad, which was 4N, and in the N, you are going to have 4 1N cells for each chromosome. And that is where meiosis is different because it is reductive. So spermatogenesis follows the full steps. It produces 4 haploid sperm, and it's produced constantly from puberty on. The testes produce about 100 million viable sperm every day. But oogenesis, the production of the egg, is very, very different. So the embryo, girls are born with every egg cell they're ever going to have. The primordial germ cell oogonium process. The primary oocyte is present at birth, but it's arrested in prophase 1. So at puberty, the various hormones that regulate meiosis will begin to complete the process of meiosis 1 and begin meiosis 2. But you'll notice that all of the cytoplasm, all of the organelles, go into the secondary oocyte. The nucleus is discarded as a polar body because it's not needed. And that's going to happen again after the sperm fuses, and you'll release the final process. So you produce 1 egg with 4 times the cellular machinery, which is very useful for the early division of the embryo. So where does the actual variation come from in sexual reproduction? Well, crossing over is the main one. When those sister chromatids, homologous pairs, come together to form the tetrad, they overlap in prophase 1. So it forms a process structure called the synaptonemal complex. The synaptonemal complex will literally be an overlapping of the chromosomes, and these chromosomes are fairly fragile, and so where they cross over, they can break. The crossover points are called chiasmata. So those breaks will reattach in random ways, creating a major source of variation. So if you look at the blue and the red in this picture here, the non-sister chromatids are held together during the synapsis process of the synaptonemal complex. And through prophase 1, they can break and overlap and reassemble. But they can only do this during prophase 1. So when you actually look at the cells separating, like in anaphase 1, we see that the red and the blue have mixed, creating a major source of variation for the final daughter cells. When you look at other sources of variation, we find, well, there is maternal and paternal copies of each gametes, but each gametes do not have the same chromosome. The alleles that you get from the mother and the father are going to be different, and the equal probability that male will be on the left and female will be on the right, and vice versa. So when you actually look at this process, you can create very different cells just by arranging them differently. So that independent assortment creates 2 to the 23rd different combinations for a human cell, for example, because you have 2 likelihoods for each of our 23 pairs. You combine random fertilization with crossing over and independent assortment, and you are nearing near infinite variation combination between any 2 people. So sexual variation has many sources of variation, including the 3 here, as well as, of course, mutation. So again, key ideas. Mitosis, 1 cell becomes 2 cells. They stay diploid throughout. 2N becomes 2Ns. In meiosis, you have meiosis 1 and meiosis 2. You have the cells that become haploid. So you have a 2N cell that eventually creates 4 1N cells. That is the fundamental difference. Now that we have a basic understanding of where the variation comes from, we have to look at how it's passed on. We have to look at the heredity of the process. Mendel, of course, studied peas. And when he studied the peas, he realized that there were different versions of traits that were true breeding, which we would now think of as homozygous. And from this information, we came up with some basic ideas. Now, I'm going to replace his terms with the terms that we currently use now, but we realize that traits come in alternate versions or alleles. These alleles vary in sequence at specific gene locations or loci. Traits are inherited as discrete units, chromosomes. Some traits are capable of masking others, and that's the so-called principle of dominance. But we do find that most genes are not linked, or at least Mendel did. He might have been wrong, but he thought so. So that means that a gene that is not linked, however, is going to either be on separate chromosomes or so far away from each other on the same chromosome that they are unlikely to be inherited together. When you actually look at the versions, phenotype is going to be the physical expression, the actual traits that are observable, where genotype is going to be the actual pattern of inheritance, whether it be homozygous or heterozygous. So looking at the ratios and looking at the ideas, we realize that the different alleles are fundamentally different in DNA sequence, and some of those are going to produce functional products, like in this case an enzyme that leads to color formation, and others will not. So having a base understanding of phenotype and genotype is fundamental to understanding how DNA sequence affects organism structure. When you actually try to understand patterns of inheritance, pedigree charts can become extremely important to our understanding. In X-linked recessive traits, for example, you might notice that the traits mostly affect males because males have only one X chromosome. Any male that inherits the disease will be affected by it. Females need 2 copies, making it far less common. So something like hemophilia or colorblindness. Mitochondrial DNA is inherited through the maternal line because everyone has mitochondrial DNA from their mom. The sperm does not carry any into the egg, so all of it comes from the mother. And so we can see that if the mother does not have the trait, it cannot be passed on to her offspring. And then you also have things like Y-linked inheritance, which is when only males are affected. There is only father-to-son transmission. It should be a fairly easy one to identify. Just look for all of the males in a particular lineage to show a trait. Something like the hairy ear rim phenotype that's seen in some Indian families would be an example of a Y-linked disease. Punnett squares can be very useful for predicting the future or predicting the patterns of relationship, but due to dominance, an organism's phenotype may be hard to determine. You can use a test cross to determine unknown genotypes by breeding it with a true breeding homozygous recessive. So in this example here, we have 2 possible results, either 100% purple, which means that it's homozygous dominant, or a 50-50 split, which means that the unknown is heterozygous. When you actually look at these processes, we come into Mendel's second principle, or the principle of segregation. When gametes are produced during meiosis, homologous chromosomes separate from each other, and each allele for a trait is segregated into separate gametes, so we can take advantage of that, and that fundamentally is what allows a Punnett square to work. When you move beyond that, we get into Mendel's third principle, or the principle of independent assortment. In this case, each pair of alleles segregates into gametes independently. Now, this is only true for genes that are on separate chromosomes or are far enough away. So if you cross 2 traits at the same time, that would be a dihybrid cross. So the number of unique gametes is going to come from 2 to the n, where n is the number of heterozygote alleles. So in the example here, we have a fully heterozygous organism. So that means 2 to the second, which means there are 4 possible gametes, and that creates the standard 4x4 dihybrid cross that you're used to seeing. If you cross 2 fully heterozygous offspring, you're going to always produce this same pattern of 9 to 3 to 3 to 1, with 9 being dominant for both traits, 3 being dominant for one trait but not the other, and 1 being recessive for both. However, if it's not that known pattern, it can get complicated, and that's where statistics is going to come in to help us. The rule of multiplication basically looks at traits and says that there's the chance that any event can occur 2 or more different ways. We can add those up, and rule of multiplication that you have 2 or more independent events that will occur together, you can multiply those out. So if we were looking at the offspring of this cross, okay, well, what's the chance of a yellow round offspring? Well, 3/4 times 3/4 is 9/16, and that agrees with the full Punnett square. So don't actually do a full Punnett square if you can just do multiplication and addition to make it work. So you can see a full breakdown of the math here for the same cross. No, you wouldn't have to do all of this. You would only do it for the one that you needed for the multiple choice question. If they ask you for an FRQ question, they ask you to show a dihybrid cross, you'll be doing the full cross. So, sorry. Now, when you look at the rest of this section, we're looking at where Mendel was incorrect. So Mendel's principle of dominance, not all traits are dominant all the time. Many inherited traits are the result of modes of inheritance that differ from a strict dominant recessive relationship. Phenotypes can result from alleles with a range of dominance, from the combined effects of more than one gene, or from genes that have more than just two alleles with the population. So when you look at the crosses, incomplete dominance is a condition when the dominant allele does not completely mask the recessive. So plants, the red is dominant to white, pink is a blending of red and white, so that would be incomplete dominance. Codominance is a condition when there is more than one dominant allele. As a result, both alleles will be expressed in the phenotype. An example of codominance may be the color of flowers again. If you have both red and white alleles with codominance, you may have mottled or variegated phenotypes. Blood type A and B would be a codominant example. But blood type is also a difference because it is an example of multiple alleles. So in this case, we have 3 different alleles that exist for blood type, but you only inherit 2 because you are diploid, which makes blood type a little more complicated. So type A and B blood can be homozygous A or heterozygous A, and both of those are dominant. But when they're both present, both are expressed separately, making it codominant. So incomplete dominance is going to mix. Codominant, both will be expressed separately. Sometimes we have more than 2 genes controlling the expression of a single trait. In an epistasis, you can have one gene that masks another. So in this example, we have the pigment gene and we have the color gene. So if you don't produce any pigment, it doesn't matter what color the fur would have been, it is epistatically inhibited. So in this case, instead of having a 9:3 to 3:1, we have a 9:3 to 4. Because so long as there are 2 little e's in this example, there will be no pigment and you'll produce a golden lab. Other situations, we're looking at polygenic inheritance. You can have traits that are controlled by multiple genes. Skin color is a great example of this because skin color is controlled by 6 known genes. So you're producing a wide variety of variation. These polygenic traits are going to be traits that exist on this bell curve relationship. So height and the genetic component of weight and intelligence and hair color and eye color, all of these things are going to be polygenic, or many genes controlling one thing. The environment can also have an effect on your phenotype. Skin color, for example, is going to be altered if you tan. The rabbit's fur changes in response to temperature. It's not 2 different species, it's 2 different weather patterns. Hydrangea flower color changes in response to pH. Human body weight can be affected by all sorts of factors. So we find that environments can alter phenotype. Now, some disorders, such as certain types of cancer or even psychiatric disorders, are traits that are both genetic and environmental because we see evidence that they run in families, because we know we can modify them by changing the environment. It's not nature, it's not nurture, it's both. Many traits are inherited through, obviously, parental genotypes. But we find one of the broader situations are traits that are pleiotropic. Pleiotropic is when you have one gene that has multiple effects on the physiology of an organism. We find that there are many genes that are pleiotropic, things like sickle cell anemia, cystic fibrosis, acromegaly. In this case, we have a certain type of albinism and dwarfism. The genes that we have covered affect one phenotypic character, but the pleiotropic ones affect more than one. The wide-ranging symptoms of sickle cell disease is due to a single gene. When you look at the intricate molecular and cellular interactions responsible for an organism's development, it's not surprising that a gene can affect a number of characteristics. An additional complication when you look at genetic disorders is the difference between penetrance and expressivity. So just because someone has inherited the trait or inherited the disease or disorder does not mean that they're going to show the same effects or that they'll show effects at all. Penetrance is the likelihood of a gene expressing a phenotype. So an organism could have reduced penetrance, which means that they're going to have a mutation, but they don't develop the features of the disorder. Expressivity is looking at the extent to which a trait is expressed. And some organisms are going to have variable expressivity. So the symptoms that occur in different people with the same genetic condition are not going to be the same. So something like Down syndrome, for example, has 100% penetrance, it's complete penetrance, but it has variable expressivity. The amount of the severity of the effects are going to be widely different from person to person. Mendel believed that traits could not be linked to chromosomes or were not linked to chromosomes, but it turns out that that's not right. Thomas Hunt Morgan and his researchers showed us that traits could be linked to the X chromosome, which is one of the major pieces of evidence that showed that DNA was the important genetic carrier of information. Sex-linked traits are incredibly common, especially on the X chromosome. So guys only have one X chromosome, so they're going to show those sex-linked traits more often. Girls, you can't have two X chromosomes active. One of those chromosomes needs to be turned off. It has too much information, it's too much control. So it turns out that in every single cell of your body, there is a X inactivation, aka a lionization, after the researcher that discovered it, and it's going to turn it off. And that's how, say, for example, the tortoiseshell cat can exist, because the black and the orange allele are randomly turned off, creating that variable pattern. You're never going to see a tortoiseshell male. Another complexity is extranuclear genes. Not all genes can be found in the nucleus, especially the mitochondria and the chloroplasts. These genes can only be inherited matrilineally. Conditions caused by a mutation in the mitochondrial DNA have unusual patterns. Both males and females are affected. The condition is transmitted through the female. If a male has the trait and his spouse doesn't, the offspring won't have the trait. In the first pedigree, you see that when a female has the trait, all of her offspring have the trait. But in the second, this is not necessarily true. The expression of mitochondrial conditions is variable, and inside each cell there are several mitochondria, and the number of mitochondria that carry the mutation can vary. So when you look at these traits, it doesn't have to guarantee that all of the offspring will have it, but it can only be passed by the mother. And one component that we still have yet to truly understand is epigenetics. Epigenetics is heritable changes in the gene expression caused by mechanisms other than changes in the DNA. There are many different components of this. DNA methylation is a more common one. You can silence genes by adding methyl groups directly to the DNA. You can also have DNA acetylation, which can bind and cause the relaxing of chromatin, which is going to activate genes. And we also have genetic imprinting. So which parent a gene comes from turns out to matter. Epigenetics doesn't affect the sequence of the gene. It doesn't affect the DNA itself, sort of as a direct component of nucleotides, but it does affect how genes are read by the cells, and subsequently whether the cells should produce relevant proteins. Basically, the human lifespan is like a long movie. The cells are the actors, and the essential units that make up the movie. DNA would be the script. So if the DNA sequence is the words, certain blocks of these words that instruct key actions would be the genes. Okay, so genetics is screenwriting. It's a complicated analogy. Do you get it? I hope. Okay, epigenetics would be like directing. The script can be the same, but the director can choose to eliminate or tweak certain scenes or dialogue, altering the movie for better or worse. When you actually look at the crosses and you look at the capability, this is where that chi-square is going to come in. If you have a hypothesis of relationship, for example, is the gene linked or not, we would have predicted ratios. So in this example, if the genes were on different chromosomes, we would expect a 1 to 1 to 1 to 1. If the genes were on the same chromosome, we would expect a 1 to 1 to 0 to 0. We look at the actual results, we find that some organisms don't follow the pattern even though it looks like it would be on the same chromosome. Why do they do that? Well, crossing over happens, there is variation. And we can use chi-square to test that hypothesis to see if the difference is significant. So when you actually look at it, we realize that recombination is happening, creating that variation. So we can calculate the amount of crossing over. We can also look at the location of the genes on the chromosome. So we can use chi-square to find out if the difference is significant, but we can use this recombination frequency to find out how far apart the genes are. Recombination frequency is going to be equal to the number of offspring that are recombinant divided by the total offspring times 100, and that tells you the distance between any two chromosomes, which we can then use to create a linkage map. The largest recombination frequency is going to be the outermost genes, and we can find the overlaps that exist and create gene maps. We can also do this for plasmids, but remember the trick to plasmids is that they're always a circle. Now, what happens if these genes don't get passed correctly? Well, you end up with mutation. So if mutation affects a group of genes or an entire chromosome, it is known as a chromosomal mutation. Malfunctions during meiosis, aka non-disjunction, can result in gametes with an abnormal number of chromosomes. This could be something like Klinefelter's and Turner syndrome, which are both mutations in abnormal numbers of sex chromosomes or allosomal mutations. Or you could have an abnormal number of autosomal chromosomes, like Down syndrome, which you can see pictured here in the karyotype. So when you have the wrong number of individual chromosomes due to non-disjunction mutations, we collectively refer to that as an aneuploidy. Okay, so aneuploidies, you have, in this case, you have 3 copies of this chromosome creating Down syndrome. Or you might have Klinefelter's, which is the XXY, or Turner syndrome, which is only 1 copy of the X. You have wrong numbers of single chromosomes. In some organisms, especially plants, you can have polyploidy, which is whole extra sets of chromosomes. Plant breeders intentionally develop polyploids with desirable traits. For example, seedless watermelons are triploid. Normal watermelons are diploid and have seeds. By using chemicals, plant breeders can double the number of chromosomes, and then the tetraploid watermelons are crossed with a standard diploid watermelon to make triploid watermelons. And those are sterile because you need pairs of chromosomes to form seeds. So plants can survive this polyploidy. It's a tool that we can use to understand the genetics of crops, but it is not something that is survivable to an animal cell. Now, in general, a mutation is the alteration of an organism's DNA, and mutations can range from a change in one base pair to the insertion or deletion of large segments of DNA. Those large segments of DNA would be the chromosomal alterations, be it a deletion, a duplication, an inversion, or a translocation, or you could have gene mutations that affect individual nucleotides. So there is a broad difference between the scale of the mutation and what it's going to do. Chromosomal translocations are increasingly becoming a product of, or at least an interest of research, because we find that there are many different diseases that are linked to just moving the chromosome pieces around. You're not changing the sequence, but you're changing the location. Huntington's disease, for example, there's a duplication that results in alteration of an enzyme that causes the death of nerve cells. Or Philadelphia syndrome, like pictured here, it's a translocation that causes leukemia. So you don't even have to change the sequence. If the DNA is in the wrong place, it can have massive effects on the organism. These chromosomal disorders and these broad mutations are something that we're becoming increasingly aware of and looking at prenatal diagnosis. So amniocentesis can be performed between the 15th and the 20th weeks, and a sample of amniotic fluid is removed and tested. Or you could have chorionic villus sampling, which is performed between the 10th and the 12th weeks. Where a sample of the placenta is removed and tested. Both of these are going to result in karyotypes, so we can see the chromosomes themselves. Both tests are invasive, and a small percentage of cases can lead to miscarriage, but it's going to be considered in situations with increased risks. But prenatal diagnosis is a way for doctors to determine before birth whether a fetus has a problem, and is something that has to be considered more and more, especially when you are looking at family history or the age of the mother.